Tomorrow belongs to those who prepare for it today. A seed planted today takes root and grows into a tree that bears fruit in the future. Over 110 years ago, a seed was planted. Today, that seed, the idea of excellence, has grown into the University of Pretoria, South Africa's largest contact research-intensive university. 9 faculties and a business school are spread across 7 beautiful campuses, which are home to over 50,000 students, ready to make an impact in the world beyond university and join our global network of nearly 300,000 alumni. Future-focused, sustainably developed facilities and cutting-edge multi- and transdisciplinary research are underpinned by a desire to transform lives and have a positive impact on communities and the world. Excellence in teaching, learning, research innovation, arts and culture, and sports puts us firmly amongst the world's best universities. Knowledge is not just what is in books. It is the wisdom to apply it, to nourish and nurture the seed so that it takes root, grows tall, bears fruit, and branches out. UP plants that seed, that tiny bit of curiosity, creativity, critical thinking, hope, the desire to care, respect, help, and innovate against all odds, to grow, to leave your mark, to excel, to challenge the norm, to think, to rethink, to discover, to inquire, to lead, to have courage, to make a difference, and to persevere. This is the University of Pretoria. We make today and every day matter.
Good day to all the attendees in the Senate Hall, as well as all online attendees. Colleagues, ladies and gentlemen, my name is Norman Duncan, and I'm the Vice Principal Academic here at the University of Pretoria. Colleagues, it is my privilege to serve as the Program Director for this, the 29th lecture in the UP Expert Lecture Series. Today's lecture will be presented by Professor Marietjie Venter and is titled From Novel Virus to Global Pandemic, Understanding the Origins and Evolution of Emerging and Re-emerging Viruses. Colleagues, ladies and gentlemen, in the context of a pandemic which the world is still trying to come to grips with, the focus of today's lecture could not be more important and topical. I now invite Professor Tawana Kupe, the Vice Chancellor and Principal of the University of Pretoria, to formally welcome you to this lecture and to introduce Professor Venter.
Thank you, Professor Duncan, Program Director. A very warm welcome, colleagues. Those in the room here, we are in the minority, I see. Most of the colleagues here, over, I think nearly 300 people have registered by this time. They are virtual or online, but that's the way of the world now, and actually gets us more audiences. This is a very exciting day, I think, too, especially because of the expertise of, it's an expert lecture, so I couldn't possibly invite a person who was not an expert, right? But this is a real expert, if you know what I mean, and also on such a very important topic. As you know, the university's expert lecture series requires that every year I choose at least 2 people to speak on an interesting topic. Of course, in the research domain, we're a research-intensive university and who's an expert in their area. So this year, one of our speakers is then Professor Marietjie Venter. I can't remember who's the second person that I chose to speak later in the year. It doesn't matter. I'll be here and you will be here. So allow me, without further ado, because you haven't come here to listen to me, I'm no expert, I'm just a Vice Chancellor. Now, let me introduce you. Professor Marietjie Venter obtained a PhD Medical Virology at Wits University in 2003 on respiratory syncytial virus and postdoctoral training at the National Institute for Allergy and Infectious Diseases in the USA in 2003 on the West Nile virus. She worked on respiratory and zoonotic arboviruses since 1999. As a medical scientist at the National Institute for Communicable Diseases, you normally know it as NICD, and the University of Pretoria from 2006. She was co-director, Center for Respiratory Diseases and Meningitis, NICD, and National Influenza Center Director from 2009 until 2014. From 2014 to 2016, One Health Program Director, Global Diseases Detection Center, U.S. Centers for Disease Control and Prevention, normally known the CDC, in the South Africa office. She's a full professor, medical virology, since 2016, heading the Zoonotic, Arbo, and Respiratory Virus Program and co-founder, Center for Viral Zoonosis, Department of Medical Virology, University of Pretoria. Her group does One Health's syndromic surveillance of acute respiratory, febrile, and neurological disease of unknown origins in humans and animals and vectors, and investigate the molecular epidemiology and genomics of respiratory viruses, including COVID-19 and viruses in Africa. She has over 130 publications in this field with an H-index of 48 and over 6,000 scientific citations. She won the National Science and Technology Forum NSTF Award in 2013 for the best output by a senior scientist over a 5 to 10 year period in South Africa. She's a member of the Academy of Science of South Africa, ASAF as we know it, since 2021. She's advisor to the World Health Organization on surveillance of influenza and respiratory, or RSV, and the WHO Technical Advisory Group for Arboviruses, and the chair of the WHO Scientific Advisory Group for the origins of novel pathogens, including COVID-19. She is also the International One Health Day contact for Africa and the Vice President for Africa for the World Society for Virology since 2020. Ladies and gentlemen, I now call upon Professor Marietjie to give us her address. And Professor Marietjie, your audience in person and online.
Thank you very much, Professor Cooper. Thank you, Professor Duncan, all my friends, colleagues. Thank you very much for attending, and all the people online as well. I hope you will enjoy the lecture. Okay, so without further ado, I would like to tell you about novel viruses, or how a virus goes from a novel virus to a global pandemic, and understanding the origins and evolution of emerging and re-emerging viruses. So just a quick overview of my talk. I'm going to introduce you to emerging diseases, epidemic and pandemic potential, quickly look at the drivers of disease, of emergence, and also what the One Health approach means. Then disease X, look at zoonotic virus classified according to the transmission route and the epidemic and pandemic potential. And then I'm specifically going to focus on respiratory viruses and vector-borne viruses, because this is my field, although these are not the only ones that may fit in this, in this category. And then I would touch, as I go along, on the research that we do at University of Pretoria. Okay, so this is a map of the last, from 1981 to 2020, that was actually generated by Tony Fauci to show all the diseases that have emerged over the past 40 years. As you can see, this is a global occurrence. Diseases keep emerging internationally and in all continents, the latest being COVID-19 that was first identified in China. The interesting thing here is that most of these diseases, up to 70%, are either vector-borne or zoonotic of nature. So this brings us to the One Health approach. So One Health refers to the idea that animal, human, and environmental health is interconnected. And in order to study this, we have to work together from a veterinary, medical, and environmental side. So the theory around this was generated by someone called Karish, and he said that or most of these diseases may be circulating in the environment, and that if you can detect them early on, having ways of forecasting this, you can predict epidemics or pandemics before it actually spills over into animals, can prevent outbreaks in animals, and this will then reduce the risk to humans, and it gives you a larger time of opportunity to actually respond to a new disease coming from animals. So why does new diseases emerge? There's several factors that have become more prominent. Alterations in the contact rates between humans, domestic animal, and wildlife hosts have increased. For example, humans that go into agricultural expansion into pristine forests. This was, for example, the emergence of Nipah virus in Malaysia and Ebola in Africa. The wildlife trade, wet market, bushmeat, and export of wildlife all play a role, and here we can think of SARS coronavirus 1 and also 2, and then things like monkeypox. Global travel, which has increased significantly, it quickly spreads diseases internationally and was responsible for the spread of SARS coronavirus 1 and 2, but also pandemic influenza H1N1 in 2009 to countries across the world within 6 weeks, and then of course HIV. Contact between farmers and poultry or livestock and wildlife. Here we think of avian influenza and Rift Valley fever. Climatic events, creation of new habitats, building of dams, the El Nino and floods. Once again, Rift Valley fever and several other arboviruses, and introduction of new pathogens and vectors into regions, including West Nile, Zika, and chikungunya. And this, of course, then brings us to global warming, which allow insect vectors, for example, mosquitoes to invade new areas. Here we think of Aedes aegypti and Aedes albopictus mosquitoes that has expanded across the world and increased the range of chikungunya and dengue virus. So not all viruses will end up in being a pandemic. And if we look back over time, you can see that there were specific pathogens that caused major pandemics and had much more impact. Not all of them may be as significant now, but there's different ways that they could have been controlled. So, for example, the bubonic plague that was spread by rats and fleas could potentially be better controlled today because we also have antibiotics. Smallpox was eradicated by vaccination, but was a major cause of disease internationally. More recently, the Spanish flu of 1918. This was during the First World War and also at a time before antibiotics to treat secondary infections. But then HIV that showed up and was first described in the early 80s. This is a slow virus, so it hides away and quickly spread across the world before it was noticed, and has since caused between 25 and 35 million deaths. So this brings us then to COVID-19. At the moment, COVID-19 reported deaths stands on over 6 million cases in 2 years. So this really pales some of the other very severe viruses, such as, for example, Ebola. But Ebola has other features that makes it important that we keep a hand on that. So the WHO actually predicted disease X as being the next big pandemic just before COVID-19 emerged. And they identified a number of viruses, most of them being zoonotic pathogens that were either insect-transmitted viral hemorrhagic fevers or respiratory viruses. So insect-transmitted viruses refers to diseases that's transmitted by ticks and mosquitoes, for example. And we think here of Crimean Congo hemorrhagic fever, West Nile, that's transmitted by Culex mosquitoes, and then dengue and chikungunya. The viral hemorrhagic fevers, such as Ebola virus, that has its origins in bats, or Lassa fever, that uses rodents and causes major outbreaks in West Africa. But then also don't forget about HIV, which was actually, it's not a hemorrhagic fever, but it was spread by bodily fluids and has been one of the major pandemics of the past century. Then respiratory viruses. These are the most effective in moving around. Viruses such as SARS coronavirus 1 was initially probably from a bat, from where it moved over to civet cats and then to humans. We don't know what the intermediate host is for COVID-19 or SARS coronavirus 2, but then MERS coronavirus that have camels as a vector or as a reservoir host. And then, of course, influenza viruses. Zoonotic influenza, swine, and avian influenza virus always remains a big threat. So what makes them so different? When does a virus have a bigger opportunity to, or chance of becoming a pandemic? So here we have to think both of the transmissibility. So how easy does it transmit between people, and how many people will one virus infect, versus the severity. So for example, bird flu or Ebola is very severe, but it's not as transmissible like COVID-19, for example. So COVID-19 only had about a 2 to 3% case fatality rate, but it caused over 6 million deaths because it could spread so far and wide. Okay, so then I'm going to look at emerging and re-emerging respiratory viruses with epidemic and pandemic potential. So there's many respiratory viruses that's been described in the past 20 or so years. They can be divided into groups that specifically is zoonosis, so they had come directly from animal to humans. This includes SARS-CoV-2, MERS, and SARS-CoV-1, the first one, the avian influenza viruses, but then also viruses such as Hendra and Nipah virus. Then some viruses that was just discovered to be more severe than what we knew, that we thought only causes a common cold, but can actually cause more severe disease. And then several viruses that were newly discovered because of improved technology, so the molecular era that made it possible to identify them, but some of them actually also came from animals. So firstly, influenza, which forever remain the looming zoonosis and potential for zoonotic diseases. So most influenza strains actually comes from wild waterfowl. So you can find all 16 H1 or H1 to H16 types and N1 to N9 in waterfowl, from where it then spread to different species. Not all of these types can infect all species, but for example, humans are sensitive to the types H1, H2, and then to a lesser extent H5 and H7 that can be also go over to birds. Pigs, for example, are also sensitive then to these specific strains, but can also become infected by bird strains. And this has several implications for creating new viruses. So in general, bird flu viruses or avian influenza viruses in humans cause mild disease, but there's a couple of them, for example, H5N1 and H7N9, that can actually cause quite severe disease and have a case fatality ratio of about 60 to 70%. However, there's very few cases because it's not that transmissible between people. So although you may get infected from the bird, human-to-human transmission is limited. So most influenza viruses have point mutations, which we call genetic drift. So that's what causes these viruses to be able to cause a new epidemic every year. But then you get occasionally antigenic shift. So antigenic shift refers to when, for example, an avian virus infects a pig, and it also gets infected by a human virus. Pigs then are a mixing vessel where you can create a new virus. And now this virus has the outside of the avian virus and the inside of a human virus. So it's very transmissible between humans, but the human population have never seen it. And that's how a pandemic happens. So the major pandemics happened every 20 or so odd years. The biggest one, of course, was the Spanish flu, which was H1, N1 viruses, then H2s, H3s, the Hong Kong flu, and finally in 2009 we had H1N1 pandemic virus. This virus originated from swine, where you can maybe remember that it was initially called swine flu virus, but it also had birds' genes in it. So all of these eventually become seasonal flu. And seasonal flu can still cause between 290 and 650,000 deaths worldwide. But in general, the case fatality ratio is low. But once again, because it's so infectious, so many people can spread it, and you get so many deaths. Okay, then moving to the coronaviruses. So most coronaviruses actually have their origin in humans. So the most recent ones that emerged was, like I said, SARS coronavirus 1 in 2003, MERS coronavirus in 2012, and then SARS coronavirus 2 in 2019. All of them had a precursor virus in bats, but then had an intermediate host. So, for example, SARS was a civet cat, camel for MERS, but we don't know yet what it is for SARS-CoV-2. So these viruses falls in the beta coronavirus group, and they can spread between humans and different animals that are also sensitive to them. In the genome of the virus, the spike protein is the surface protein, and this is where most of the changes occur, and it's also responsible then for binding to the receptor binding domain, or binding to the receptor in the human cell to be able to infect humans. So if you compare those viruses, the first one identified, SARS-CoV-1, had a fatality ratio of 9.6%. Middle Eastern virus was identified in 2012 and had a very high case fatality ratio of 34.4%, while novel H1N1, coronavirus, 2019, actually had a fatality rate of about 2.1%. However, this slide comes from early to 2020, and you can see back then there were only 304 deaths, and now we sit on over 6 million. So it doesn't necessarily mean that the virus that has the highest fatality rate will cause the most deaths. One of the features of all of these viruses is an incubation period of 1 to 14 days, but COVID-19 was able to spread to different patients or different humans before it actually becomes symptomatic. So before you know that the virus had infected people, it already spread widely. All of them have fever, cough, shortness of breath, and diarrhea in mild cases, but then in severe cases, patients develop pneumonia, severe acute respiratory syndrome, kidney failure, and death. So specifically looking at COVID-19, there were some viruses identified in bats that had a precursor virus that is very similar to it. So there's one that's called rat G13 that was identified in 2013 in China, that was about 96.1% identical to SARS-CoV-2, and another one that was more recently identified in Laos, which is on the border of China, that had 96.8% identity. However, this is still quite far from the actual SARS coronavirus 2, and we know there should be an intermediate host, but so far it hasn't been identified. Pangolin was actually less identical than the bat coronaviruses, only 90%, and we haven't identified anything that's closer to it yet. So initially when it was discovered, many patients had a link to wet markets in Wuhan. And in the wet markets, they sell lots of different animals. These animals were tested, but although surface contamination were identified that tested positive for SARS-CoV-2, none of the animals actually tested positive. After it then spread to humans, it really became a human pandemic. So animals had limited to no role played in the actual pandemic spread to humans. And it was really vaccination that will eventually solve that, or herd immunity, but it comes at a higher cost. But what did happen that was quite interesting is several reverse zoonosis cases was identified. So animals became infected from humans, and this gave us some clues into what could potentially be the intermediate host, but also create a risk of potentially creating a new animal reservoir. So many of the natural infections was actually domestic animals or animals in zoos. And so in zoos, it's an artificial situation, but there has also been cases where it spread amongst different animals. So, for example, farmed mink was actually the most susceptible to COVID-19, and during the pandemic, they had to cull out hundreds and thousands of minks in Europe because minks actually spread it to each other, and they also could spread it onto humans. Other animals that also were shown to be able to potentially spread it between themselves is white-tailed deer, and then also hamsters. So there was an outbreak in a pet shop in Hong Kong of hamsters, and the hamsters actually could infect the pet pet shop owner. So far, if you look on the World Health Organization for Animal website, or the OIE, there has been over 600 outbreaks reported, so 675 internationally. And you can see in South Africa, we actually also identified cases. These are the only ones on the African continent. And so this is where our group was actually doing surveillance for respiratory diseases in animals. And we work with a wildlife vet from the Onderstepoort Veterinary faculty, as well as with Dr. Malumba from the Onderstepoort Veterinary Institute. And we identified some lions, first a puma actually in the beginning of the pandemic that tested positive for COVID-19. And then during the Delta wave, we identified 3 lions in a private zoo in Johannesburg. One of them developed pneumonia and actually was quite sick, while the other 2 had some difficulty in breathing. But they were in different cages. So when we then did a One Health investigation to test the people that work in the zoo that could potentially have contact with them, we identified the big cat keeper, that's actually the guy that feeds them and cleans their cages, that was positive by PCR for COVID-19 at that stage. And all of them were sequenced. We sequenced the genomes, and they all had the identical Delta variant. What was also quite interesting is the lions stayed positive for up to 7 weeks. The zoo had to be put under quarantine for that period, and we kept testing them, and they were positive for that long, but then they cleared the infection completely. So they are not carriers. They will clear the infection, but the risk is that if it is in a wildlife environment, it could potentially spread further. So it's important to actually increase surveillance in wildlife in South Africa as well. So where do we stand at the moment? So internationally, there's been over 518 million cases identified or reported of COVID-19, officially to the WHO, and over 6 million deaths. If you look at the epidemiological curve from when it started, you can see we've had these different waves. So there were basically 4 waves. And at the moment, the wave is actually quite significant. It's the highest. But what was also interesting is you can see the death rate actually is coming down at the moment. And Europe and the USA feature much higher in these outbreaks, and the little blue stripe that you see there is Africa. But of course, this is likely because of underreporting. So we don't test as well as they do in Europe and the Americas. So recently there's been a study that looked at excess deaths internationally, and what they found actually is looking in about 250 countries, the total excess deaths was 18.2 million. So it means that 18.2 million more people died during this time than what would normally die in previous years in the pandemic. So it shows that it's highly underreported. So coming to South Africa, you can see we also had our 4 waves, and also not to forget about is the hospitalization of people. A very significant number of people that were hospitalized in this time, but we reported 3.8 million cases and 101,000 deaths. Once again, when the MRC looked at excess deaths, they identified 309,000. So it means if you look at the reported cases, that's in red here, versus the actual cases or the excess deaths, there's at least a 3-time underreporting. Okay, and then towards the end of 2020, we started seeing these variants occurring. So what is driving these different waves that we saw? Initially, COVID-19 seemed to be quite stable, and although there were some point mutations, basically the world was naive and it didn't really adapt yet, it didn't change, so it came from animal hosts and it infected people, but as it moved along, it improved itself. And so the area around the receptor binding domain, the ACE2 receptor, is especially the area that will change. And then we identified, or WHO identified these variants that they call variants of concern. They have increased transmissibility, increased virulence, and decreased effectiveness of public health measures such as some of the vaccines and diagnostic tests. So the first one that was identified was the alpha variant in the UK, then the beta variant in December when all the kids went to Margate. Do you remember that? So in 2020. And then the Gamma variant in Brazil, Delta variant in India, and now recently Omicron in November 2021 in South Africa. So I'm fortunate to be part of the National Genomic Surveillance Network. So we are a group of labs from across the country. It includes academic labs in all the universities such as UCT, Stellenbosch, the NICD, University of Pretoria, Witwatersrand, and CRISP, or University of KwaZulu-Natal, and University of the Free State, that work with the NHLS labs in those, the National Health Laboratory Service labs in those areas. And we basically collect the diagnostic specimens that test positive for COVID-19 and submit them for genomic sequencing. So through this program, we identified first the beta variant, then showed that the beta variant was replaced by the Delta variant, and then unfortunately identified Omicron as well. So in the end of December, or in the end of November, we saw that there was one of the tests, the PCR test, that one of the targets, so we called it the S gene target PCR, stopped working. And it was actually first in the private lab that they noted this. And when those specimens were sequenced, they noted that this is a new variant. So it started in Tshwane. So we was the epicenter of the initial outbreak. And from there, it spread on to the rest of the country very rapidly. What's interesting, if you look at the phylogenetic tree of, of the different coronaviruses, you can see this is the root of the tree. So from 2020 in March, when we initially started seeing cases, through to 2021 in September, you can see how the virus had changed. And here you can see the different variants. And what was interesting is Omicron was not closer to Delta. So if you think of that first picture that I showed you of flu, it wasn't a drifting out of Delta. It was completely different. So it had potentially done a shift. So it was different to the original one. So over the genome, there was about 52 amino acid differences. Within this spike protein, 26 to 32 differences, and then within the receptor binding domain, there were 15 differences. And we know some of these were similar to some of the other variants, and we know it leads to increased transmissibility, improved binding affinity, and enable the virus to escape some of the neutralizing antibodies. Some of them are new and specific to COVID-19, and we know that some of them make it more infectious, and to Omicron. It makes it more infectious and make it harder for antibodies to attach or kill the virus. And then there's a number of mutations that we don't know what it does yet. So this has been an unbelievable network of South African collaborations. We worked with the pediatricians at Kalafong and Steve Biko Hospital, and we actually showed that the Omicron variants were more or led to a higher incidence of hospitalization in children. We worked with Penny Moore at the NICD, and she actually showed then that infection of vaccinated individuals with Omicron actually caused or resulted in a very significant antibody response, neutralizing antibody response, while unvaccinated individuals didn't have such a great response. So being vaccinated gives you a better antibody response to a wider range of different variants, while if you weren't vaccinated, you weren't as well protected. So even though there's escape, we also showed then with Wendy Bergs at UCT that the T-cell response was also still very significant, and it still protects against COVID-19. So at the moment, although you can get reinfected, there's many reinfections, the vaccine protects against severe disease and death. Also through the NICD and the Network for Genomic Surveillance, they showed that COVID-19 Omicron variant was much less severe. So if you look at hospitalizations in this fourth wave relative to the previous waves where this S gene target still worked, it was a 2.4% hospitalization rate relative to 12.8% in the previous waves. Shabir Mahdi then also did a study where they looked at the the seroprevalence in Gauteng, and they actually showed in unvaccinated individuals there was up to 68.4% antibodies to COVID-19, while in vaccinated individuals 93.1%. So this could relate then to the uncoupling that you see with severe disease with Omicron. So it may not only be that the virus is less severe, but because we have such a high level of immunity. And then over the last couple of weeks, we saw that Omicron started changing again. So the initial variant was the BA1 variant, and then we identified a couple of mutations or different variants early on, but then they started taking over. So first the BA2 took over, and then most recently we saw these BA4 and BA5s. So basically you can see the one variant is replacing the next one, or subvariant. Sorry, it's not new variants, but it's subvariants or different versions of Omicron. And finally now with B4 and 5. And once again, there's significant mutations around the receptor binding domain. And also just there, you can note they also actually started in Gauteng, and from there it spread across the rest of the country. So what we see at the moment, the upsurge of cases, is really driven by this BA4 and 5. So where does this weird virus come from? Okay, so you can see it's this weird long branch that doesn't actually cluster out of any of the others. So what the theory is at the moment, or there's a couple of hypotheses. Firstly, potentially, could it be an animal source? So could it have been a reverse zoonosis that have mutated and gone back to humans? Could it have evolved just in a population that's under surveillance? So in South Africa, we have quite good genomic surveillance, but in other parts of Africa, we don't. And we know that we have one of the busiest airports in the continent, so some of those patients would not fall under our surveillance system. But then also, what about chronically infected COVID-19 patients? So I'll tell you a little bit more about that, but they actually can develop significant mutations as well. And then low vaccine coverage in Africa is a fertile ground for emergence of new variants. So is it possible for variants of concern to involve in animals? So shortly after Omicron was identified, this group actually described that the mutations that was identified in Omicron was very similar to mutations that develop if you adapt the virus to infect mice. So, and they tested this basically by looking at if the mutations in that region would make it more infectious for mice, so it binds better to the receptor binding domain, and then also to humans and lesser extent to other animals. Then a group in New York that does wastewater surveillance saw that in the wastewater there's some lineages that is not seen in the human population. And then when they sequenced the water and look at the DNA sequences in the water, they could see that in the water you find human DNA, cow, pig, rat, dog, cat, and sheep. And they then built these mutations into pseudoviruses to test it, and they could also see that it actually then binds better to mouse and rat and human. So this is all theories at the moment. It's not proven. It hasn't been found in nature yet, but it gives us some clues of where to go and look. And then interesting, you may have heard in the news that the white-tailed deer in the United States have been identified or been infected with COVID-19. So one of the theories is this is the hunting season, and also when the COVID-19 outbreak was most severe, the deer got infected, but then as it reduced in the human population, it just kept on spreading between deer. So they're not getting reinfected every time by humans, they are spreading it amongst themselves. And then in Canada, they identified this also weird long strain that from a deer that actually had evolved as well in deer, and it was able to infect a human. So at the moment, farmed mink, pet hamsters, and white-tailed deer has been shown to be able to infect humans with SARS-CoV-2. This is not to say any of these are a potential reservoir, but it reminds us that it is important to actually go and look if there potentially could be new reservoirs. And this resulted in the OIE, WHO, and FAO releasing a joint statement to ask for the monitoring of SARS-CoV-2 in wildlife and preventing the formation of animal reservoirs. So then what about the HIV or immune suppressed patient? So Dr. Karim and his group actually showed that HIV patients that was not, that treatment failed, so they were not controlling the HIV well, could actually become infected with COVID-19 and stay positive for up to 90 or longer days, so 300, 200 days here. And what happens then is this patient still get antibodies, so they get exposed to different coronaviruses, they develop antibodies to that, but then they evolved and actually generate then this new variants. So they don't actually clear it like a healthy person would have done. And this could then potentially lead to a significantly mutated variant. Okay, so then moving to emerging and re-emerging vector-borne viruses with epidemic and pandemic potential. Vector-borne viruses, referred to as arboviruses, are viruses that's transmitted by insect vectors, as I said before, and normally have an animal host or reservoir, and then can also infect people through mosquitoes or through direct zoonotic transmission. So typical symptoms is fever, headache, myalgia, arthralgia, morbilliform rash, and conjunctivitis. But then in some cases, you'll get more severe disease, such as encephalitis in the case of West Nile, paralysis and hemorrhagic fever signs, dengue, and then also in animals, you can get some signs such as abortion with Rift Valley fever, encephalitis, and hemorrhagic manifestation and death. So if you follow animals, you're able to identify these outbreaks happening if they are also sensitive to that. So there's various different families involved, and in Africa there are specific viruses or arboviruses that's well known. So chikungunya and Sinbis virus, both transmitted by mosquitoes, but one by Aedes and Culex mosquitoes is associated with a rash and arthralgia. Then Zika virus, West Nile virus, and measles-Brown virus can all cause encephalitis and are also transmitted by Culex mosquitoes or Aedes mosquitoes. And then yellow fever, dengue, and Rift Valley fever in Crimean Congo are what we call viral hemorrhagic fever. So they can potentially cause hemorrhagic fever signs, with all of these being transmitted by Aedes mosquitoes. Only Crimean Congo is transmitted by Iloma ticks. And then other lesser-known viruses, such as Shuni virus, that I'm going to tell you a little bit about, have various hosts. They also use culicoides midges that can spread them between animals, and then different mosquitoes that can allow them to infect humans. And they cause neurological disease and can also cause abortion. But apart from these well-known ones, there are several others around, okay? And so when you start looking for them, you realize that there are several arboviruses in Africa that can cause similar signs, but are located here. And so it's important to also look for them, because these could be the potential emerging viruses. So there's been several emerging arboviruses from Africa that had caused international outbreaks. So we think of West Nile virus that spread in 2000 to the Americas, and from now can actually be found almost all over the world. Zika virus also originated in Uganda, spread to Asia, and then on to Brazil, where it caused microcephaly in children. So it was only noted at that stage. And then chikungunya virus, it's an African virus associated with arthralgia that spread on through the rest of the world. So why do you not find all these viruses all over the place? So if you think of these viruses, chikungunya, dengue, and yellow fever, that are transmitted by Aedes aegypti and Aedes albopictus, you can find them in areas around the tropics. Also, chikungunya is found around these areas because they are transmitted by these mosquitoes that are more adapted to tropical areas. While the Culex-borne viruses that have bird reservoirs, such as West Nile virus and also Sinbis virus, you can see can actually spread to other areas, because Sinbis Culex mosquitoes can also survive in cooler areas, or found in cooler areas. And then viruses such as Rift Valley fever and CCHF, or Crimean Congo, that are associated with the animal host, is more found in the area where you would find that specific host. So you can divide these viruses in two groups. The first one then is arboviruses that use non-human primates as a natural host in a salviatic cycle with endemic Aedes mosquitoes. And then in an urban cycle, it can be picked up by this Aedes aegypti and Aedes albopictus, where it can be spread internationally and cause large outbreaks because it only needs humans as the only reservoir to be infected. And this is then yellow fever, chikungunya, dengue, and Zika virus. So chikungunya is a good example. So in Africa, it will circulate between some monkeys or non-human primates and mosquitoes, and so you'll find these endemic outbreaks. But when Aedes aegypti emerged across Africa, and also Aedes albopictus, it then got picked up by this. It spread through the Indian Ocean islands up to India, and India got picked up by Aedes albopictus and actually caused over 1 million cases. From there it went on to Italy and can now be found all over the Caribbean as well. Okay, so this is the range of Aedes aegypti mosquitoes. So you can see it's in the tropics, and this is then where you can find these viruses. And Aedes albopictus is the Asian tiger mosquito, then expand the range further. So in Africa, yellow fever, Zika, and dengue virus is not found in South Africa. You can see it's more in the tropical areas. Chikungunya, there's been some outbreaks in South Africa, but this is more related to the sylvatic or the urban cycle, the sylvatic cycle, and not to the presence of Aedes aegypti mosquitoes here. The second group of arboviruses are then arboviruses with bird, ruminant, or wildlife reservoirs, and only spills over onto humans occasionally. So it will be spread between the wildlife or ruminant hosts, so Wesselsbrand and Rift Valley fever, that's transmitted by endemic Aedes mosquitoes or floodwater mosquitoes. And then West Nile and Synbus virus circulates between birds and Culex mosquitoes, and then occasionally during times of high rainfall, it will spill over onto humans and also to other sensitive animals such as horses, where it can cause severe disease. So if you look for cases of neurological disease in horses, you can also see where there should be cases of human outbreaks. So West Nile is spread all across Africa, but it's really very much underreported. While, So while Rift Valley fever occurs during large intervals, so only every 20 years or so you get very big outbreaks in South Africa. So it's not here, present all the time, while West Nile you can find almost every year. The reason for this is then also that West Nile is transmitted or is carried by birds, so it has basically the ability to fly all over the world, and then there's local mosquitoes in these areas where it could be picked up. And that's how our group actually started investigating West Nile, because this lineage 2 virus that occurs here also eventually emerged in Europe, and we could show that we have horses and human cases that's all associated with this lineage 2. So at the University of Pretoria, we have a One Health program to look for zoonotic arboviruses. So we use surveillance in animals, where we look for bird fatalities, neurological signs in horses and other wildlife, and then also if you see abortion. And then we do human syndromic surveillance, where we look for febrile disease, neurological and arthralgia signs, and then at the NICD, they'll also look for hemorrhagic fever signs, for example. And then we also do vector surveillance. So the group started basically back in 2008, and that's, as you have heard, I initially worked on West Nile virus, and so after returning from the United States, I wanted to know, why don't we see more West Nile here? And because horses are all imported to South Africa, our theory was that if the viruses that we have here is as severe as the ones or pathogenic as the ones in the United States, we should also see neurological cases in horses. So my friend, Dr. June Williams, that sits here and I'd like to welcome to the talk, was really my partner in finding West Nile. And we first started our first case, we just talked about it, the horse was in Junction back in 2008. And then every year since then, we were able to show that West Nile circulated here. But we also test for those other viruses. So we test for Flavi, Alpha, and Bunya viruses, and we could show that there's other viruses that co-circulate that was not very well known. So we can find Middelburg and Shuni virus every year as well, although they may not be as common as West Nile, but Middelburg is quite common. And also then Sinbis in horses that was not known to be a pathogen of horses. So we could show that the arbovirus season is basically between January and the end of May, and most of the cases occurs basically late summer and autumn, which is the rainy season in South Africa. We could show severe disease occurs. West Nile had a 36% fatality rate in horses, while the Shuni virus 44%, and all of them had over 94% of cases had neurological signs. You can see this is paralysis in the horses. This horse is from Gauteng, and this is also a horse from Gauteng whose tongue is paralyzed, and this horse is completely paralyzed. We also identified it in various wildlife species. During the lockdown, we actually found flamingo in the Pretoria Zoo because they didn't clean out the dam so well, and there were lots of mosquitoes, and so they died of West Nile virus. But they were exotic flamingos. Then to look at humans, we first did a sero survey in veterinarians across the country, so the same veterinarians that send us cases. We have about 150 vets that send us cases on a daily basis, and we could show that firstly in animals, all these viruses were distributed all across the country, and veterinarians had antibodies sort of in the same areas as where you found horse cases. So we established then active surveillance in hospitals in Gauteng and in Mpumalanga. We found West Nile in hospitalized patients with febrile disease of unknown origin and neurological signs. About 90% of them had neurological signs, and it accounted for 8.3% of unsolved neurological and fever cases in hospitalized patients. And it clustered with all the cases that we found in animals, so most of them were lineage 2, but we also found 3 lineage 1 cases in a lion in the Kruger Park and then 2 horses in the Cape. We also found Sinbis. Sinbis is thought to be a milder virus, but one of our students, Keiji, she's here in the back, she actually also found Sinbis virus in 12% of cases. And surprisingly, a lot of them had meningitis as well. Then other alpha virus, Middelburg virus, was more rare in humans, so we found that in 2% of cases. And once again, these alpha viruses were similar to the cases that we found in animals as well. And then finally, Shuni virus, we found in 5% of cases, and we only do PCR at the moment. If you add serology, you would likely find more. And some of these patients one was in a baby of only 13 days old, which means it's probably transmitted from the mother to the baby. It never left the hospital. And they all had neurological signs. And again, Shuni was common in several wildlife species with paralysis in horses, and also then similar to the human strains. So we then did some vector studies. So we went to areas where we found animal cases. We established surveillance sites in Boschkop, which is east of Pretoria, and Kailami, and then 2 sites in Limpopo, in Marakele and Lapalala, and also then in Mpumalanga, close to the Kruger Park, and in northern KwaZulu-Natal. In our long-term surveillance sites, we could show that Culex mosquitoes are the most common mosquito in all of these sites, but you also find floodwater Aedes mosquitoes. And then, interesting enough, if you go and look closer at the Aedes, in Gauteng we could find Aedes aegypti being common in both Boschkop and Kailami, but not in the 2 Limpopo sites. We then screened them for these different arboviruses, and we could find West Nile, different flaviviruses, Sinbis, Middelburg, alpha, and other alpha viruses, and then Shuni virus in several different species of mosquitoes. Okay, so to conclude, disease X is likely to be the emerging or re-emerging zoonotic or vector-borne virus. How do we know if it has a pandemic potential? Usually it will be a new virus to a naive population, but it can also emerge just into new regions. The transmissibility and human-to-human transmission is an important factor. And then if it can spread asymptomatically, the chances are bigger that it will spread before you realize that. And then the disease severity will determine how severe the pandemic is. Respiratory pathogens, influenza always remains a high threat, and surveillance in humans and animals for emerging subtypes is very important. The coronaviruses, COVID-19 was the most important pandemic virus of the century to date. It likely originated in bats, but we still don't know what the reservoir host is, or the intermediate host is, sorry. And the human population was completely naive to this virus. Case fatality rate was only 2%, but it spread asymptomatically, and almost all humans across the world have been exposed. So that's why we have such a high fatality, or high number of fatalities. It adapted through the pandemic to become more infectious by binding to the ACE2 receptor of humans. This made it more transmissible, but also several susceptible animal species have now been identified. That it can evade the neutralizing antibody response. The T-cell response seemed to be more consistent. Vaccines against the dominant epitopes gives us more protection than being infected by the different variants by itself. And then the origin of new variants. So is this because we don't have enough surveillance and it's just emerging in different human populations? Possibly reverse zoonosis or animal host. Immune suppressed patients, it's important that we ensure that immune suppressed patients actually have control of their HIV infections, and then it's important that we improve surveillance in animals. The pandemic potential of vector-borne viruses, it's important that the animal reservoir and the susceptible vector is present, so it needs to be widespread, or it needs a human or other host that can spread it. So the viruses, such as those with Aedes and human transmission cycles, cause large outbreaks internationally where the Aedes mosquito occur, but then also bird reservoirs can spread these viruses widely, and Culex mosquitoes that's susceptible to that in other continents can then pick it up. Culex and endemic Aedes mosquitoes is widespread in South Africa. This will ensure that outbreaks of West Nile, Sinbis, Shuni, and Middleburg virus will continue to occur through and being spread by birds. These viruses contribute to unsolved fevers and neurological cases in hospitals in humans and animals. So it's important to vaccinate sensitive animals. There's actually a West Nile vaccine for horses. And then also humans using vector control or repellents during summer. Aedes aegypti is present in Gauteng, so it's important that we have a handle on that we keep on monitoring it and practice vector control should it ever increase. Okay, and then for pandemic preparedness, I think I hope you understand that the One Health approach is very important, where we look at humans, animals, and the environment. Genomic and syndromic surveillance is useful for finding new variants and emerging diseases and virus discovery in humans, animals, and vectors. Vector control. Vaccination of humans is possible for yellow fever, and then for the animal reservoir in certain cases such as Rift Valley fever. And then the development of rapid diagnostics, vaccine development, and non-pharmaceutical interventions remain important to be prepared for the next pandemic. Okay, so thank you very much. This is my group. So the work comes over many years, and many of these students already graduated or moved on. This is my current group. Photo's a year old, so there's a few others as well. June Williams, I show there in the back, and Johan Staal has been very good collaborators over the years on animal pathology. Our entomologist was Professor Almeida from Portugal, but then also Melina and Megan that was involved in that, and then many of the other students that's been involved in the different work. The work has been funded by the GDD or CDC in the past, then currently by the EU, and then also by the G7 and the BMBF, and then local grants from NRF, PRF, and the NHLS. And then I've had many collaborators internationally that contributed to the success of the program. And then NGS SA, we've only really been doing this now for 2 years, but or a year maybe. And it's been a very good experience. Julia de Oliveira and all the other leaders from the different labs. And then all the people that's in the labs that we've been involved in that. And so thank you very much. Okay.
Professor Venter, thank you so much for that captivating presentation, very instructive. Ladies and gentlemen, I think Prof. Venter deserves another round of applause. Thank you once again, Professor Venter. Ladies and gentlemen, we come close to the end of this evening's presentation, the 29th lecture in the expert lecture series, and I'm now going to invite the Vice-Chancellor to do the vote of thanks and to close this very instructive presentation this evening.
Thank you very much, Professor Marietjie Venter. This is the University's Expert Lecture Series, and I think that everybody can agree here that, you opened our eyes to things that we thought we knew about these viruses, because in the popular media, they just talk about the coronavirus, COVID, and all of that, but they don't actually give it the scientific, historical context that you did and the dynamics that are involved, and also locating your own work and that of your group and your collaborators. So thank you very much for those insights and analysis, and also the red flags and warnings that should prepare us as a society and societies across the world. So wishing you well with your group and the work that you do locally as well as on the global stage. But thank you very much. Colleagues, thank you very much for coming to this expert lecture, those in the room and those online. The next lecture, I'm informed, is by Professor Marietjie Venter. Interesting. He's also done some work related to what you do, modeling with the Gauteng province during COVID. But I don't deem to know what he's going to talk about. But again, Prof. Marietjie, thank you very much, and power to your intellect. Thank you.
We all see the same world, but research creates a new vision of what is and what is possible. Research is central to the University of Pretoria's vision. We strive to expand the boundaries of knowledge, to develop thought leaders, to answer global questions, and to create an impact that can change lives. A combination of high productivity and excellence has earned UP an international reputation for research that is a game changer for Africa and the entire 21st century world. What we do will shape the future. The University of Pretoria's diverse research platforms offer a launchpad for today's generation of knowledge seekers. Unlocking tomorrow's potential in the unknowns of today. Our research themes and priorities pave the way to a more sustainable and equitable future for Africa and the world. Covering topics from food security to astrophysics, health and well-being to new business, and from biodiversity to education in the digital era. Engineering 4.0 is a platform of synergy for the development of smart transportation networks where digital technologies and augmented intelligence will enable new communication networks and urban development. The ever-evolving art of Africa has found a new home at the Javet UP Arts Center, where the deep histories and cultures of African people combine with technology to inspire new creativity. Recognizing that knowledge cannot be compartmentalized, UP champions a transdisciplinary approach. Creating Future Africa, a hub, a community, a home for African scholars, and a meeting place. A place where digital connectivity enables minds to meet without boundaries, and shared knowledge enables new solutions to African challenges. Welcome to the University of Pretoria, where we do research that matters.